Okay, so first of all, the last day of classes is this Friday, and at noon there'll be a kickball game outside. I think the weather's going to be decent, and it's between chemistry and biology. And so everybody's welcome, encouraged, and asked to come play or watch. And you can play, and you don't have to worry about if you're going to just come out and have some fun on last day of classes. And we'll, as usual, just be to the chemistry department. Okay, so this is where we're at. I will hand back your exams at the end of class today, and then I'll send out a link to an answer key video. And the answer key video starts with anonymous distribution of scores, so same as our first exam, where you can see how the class did overall, and where you did, and what your grade is. So on your exam that I'll hand back at the end of class, I've just marked things wrong, and then you have a circled score at your exam. And in the answer key video, I explain what I saw in the exams and what some of the right and some of the wrong answers are to try to help you learn more. And so everybody should watch that, no matter what your score was. And then there'll be this grade distribution, and you'll be able to see your score. It'll be a little asterisk next to a number, and there'll be a grade by that. And this is a hard class, and students are not doing as well as they would want to, or maybe as well as they have in some other classes, but everyone recognizes this is a hard class. And student scores are going up, and so that's good. Everybody is in a good position to get a decent grade in this class, and not their favorite grade in some cases, but a good enough grade like that. But we really have to struggle to do well on the final. So the first thing is to go over your second exam, because the final exam covers that same material. And it's this style of asking and answering questions, these two-point questions. And the final exam will cover all our material, starting with those play model videos like that. And then include this last chapter we're going to do, the chapter 11, the nucleus. So we're going to cover that this week, and we'll have no problem covering it. It's actually a pretty short chapter, and there's some high points that we want to get out of it, and we'll include those in the exam. The chapter 11 won't have a special focus on the final exam. We'll cover everything on the final exam. And our course has been building up the whole way. And we are not going to ask you to do any sort of, well, now let's apply our knowledge. Just what is this? What is this? What is this? To check and see how well you understand principles, concepts, terms of our course. So then on Thursday, Riley's going to come, and he's going to talk one more time. He's going to talk to us about really the overall story of T-cell therapy and how it's done with the patient, and then future of it, and potential to apply to other medical conditions and so forth. Our exam is a week from today. It actually starts at 8, like that, 8 a.m. It's in this room, and same format. You'll have plenty of time to answer these questions, and it covers the whole course. So that's where we're at. And do people have questions about anything at all? I have time. We can schedule time to meet this week, and I don't have anything on Monday, so we could meet anytime on Monday as well, like that. I'll be looking for questions over the weekend, like that. If I get a bunch of questions or some good questions, I make just a little tiny video and send that link out saying, let's just make sure we understand this concept, because students are confused by it. So keep your eye open for that, but it's unlikely. And we can answer questions by email. We can schedule times to meet via Zoom or in person, like that, meet with you or other students as well, like that. Anyone want to do that? And we have time to talk about stuff. Like I said, we'll put some new stuff in this week, but it's not a big chapter, and it has some good stuff in it. And we've been talking about the nucleus, so it helps us with that. I just want to show you the overall structure of our course. And so what we did is the book has these 4 parts. And we picked some background and intro chapters out of here to really get us going, like that. So those are covered on the exam. And then we jumped to this, and we were trying to get to this the whole semester, like that. And so, that was a big goal of our course. And we skipped entirely over this. We're just going to do one chapter of it. This section people would call cell biology. So I said at the start of our course, this is really 2 courses in one, molecular biology and cell biology. And so keep that in mind. Today, and Thursday when we talk about the nucleus, it's a cell biology topic. And so cell biology is less reductionistic than molecular biology. We go down to the level of cells, and especially the organelles in cells. And so cell biology emerged as a field of study in the 1950s when scientists first were able to look at eukaryotic cells with an electron microscope. And they saw organelles, and they had only seen the kind of blurry indications of organelles prior to that. So they started naming them and saying, well, what's the mitochondria do? What's the peroxisome do? What's the nucleus do? Like that. And they had some indication of some of those organelles prior to that with light microscopes, like the nucleus a little bit. But cell biology is all about that, and all about, well, what's the function of the nucleus? And so we'll look at the molecules that are regulating the role of the nucleus, and that's molecular biology. But a lot of what we want to look at in chapter 11 is the nucleus itself, and the actual physical structural nature of the nucleus and how that gives it its function. And each of these chapters in section 3 is like that. So protein sorting and transport, that's about the ribosome and about the rough endoplasmic reticulum and the Golgi apparatus. Here's the mitochondria and other organelles like that. Cytoskeleton, we talked a little bit about that, plasma membrane, and so forth like that. So they are cell biology topics like that. And so I just want you to be aware of that you know, what we've experienced is really a course in molecular biology with just very little of cell biology. And for most people, it sounds like, well, why are we really drawing a distinction? And the answer is we don't need to. But it is interesting to see that they have some different approaches, some different principles that really rise out of cell biology versus molecular biology like that. And so that's our thing, right? Where the first two sections are really introductory and general overviews, like overview of all RNA transcription, right? We cover that in chapter 9, like that. And then it dives into sort of two sections, one a section in cell biology and one a section on molecular biology that's really focused on cancer biology. So that's how we're dealing with our course. And there's a lot of information in this book, but it's actually a very well organized book. So Riley's going to come on Thursday and talk about the therapy, right? And so we really dove into the molecular biology of it, you know, designing and building the CAR gene, and then its role as a receptor, and its role as activating multiple pathways in the cell like that, signal response pathways, so multiple cell responses that the T cell normally does. The CAR is in a normal T cell. They've added it as an extra hybrid receptor. It has 3 different receptors that have been put together in its intracellular region, and they're going to put together by using the DNA for the 3 different genes. The CAR T cell has this modified receptor plus the 3 normal receptors. And in CAR T cell therapy, it's only the CAR that gets activated. The other receptors don't get activated. But the CAR is designed to activate all 3 of their signal transduction pathways to get this amplification and this integration of the response. And so Riley will talk about how that's advantageous in this strategy, how we're going to take the patient's cells out, and what we're really doing is we're making them more responsive, more ready to be activated. There's a lot of checks and balances. One of the concerns in CAR T cell therapy is these CAR T cells, they are really ready to be activated, and they could activate and damage some other healthy tissue somewhere in the body. They could start attacking the thing, and that would be an autoimmune disease problem like that. They refer to it as toxicity, that there's cellular toxicity with these, and it's on specific cells elsewhere in the body. And so that's one of the challenges of CAR T cell therapy, is to keep it limited to that cancer cell, which of course is one of the patient's own natural cells. It's just got issues, right? So how can we target that natural cell that's out of control and not hurt other tissues in the body? And the other tissues are based on the proteins they're expressing on their cell surface that the CAR could bind to, and that T cell activate and kill those cells. And we have to be careful in looking at all the different cells, because it could just be like the lining of some part of the nephron in the kidney that gets attacked. like that. So Riley's going to talk about that, and just there's lots of this on the web because there are a lot of patients and their families who are trying to understand this. So we don't have just any more reading assignment for Riley's visits than that one section and page in chapter 20 about CAR T-cell therapy. But you can solidify your learning just by Googling it, and you can find some pretty straightforward videos to help explain the picture to you. And part of that is not so much for the testable part of it, but just so you can understand it when you run into it over time, because it's a new kind of cellular therapy that you're going to see and hear a lot about over the next several years. And so I just wanted to include that. And it's this cellular therapy. Instead of trying to develop a drug, they're trying to harness the powers of cells. And we've learned a lot about signal transduction pathways and the kind of cellular responses those can give rise to. And so, doctors are trying to kill cancer cells. They're going to use X-rays and kill the cells, or they're going to use a drug and kill the cells. And this is harness the power of a cell and its capability to induce apoptosis in the cancer cells and have the cancer cells kill themselves. Okay? Here's just another image, and you don't have to look at it in any detail, but it just starts to get into more advanced concepts of that there's activation that's occurring like that. And something we didn't talk about is that there are additional receptors that are also going to try to turn the response off, because the cell is the T cell very carefully trying not to attack the normal cells. So we can activate those 3 receptors, 2 of them are shown here. But there are additional receptors that will try to turn that off, like that. So this becomes a good example of a really advanced story in receptors and cell signaling and activation of cellular responses. And this just adds here that one thing that tumor cells do is they can evolve over through natural selection to suppress the immune system, to suppress that response. So that's what's showing there is, here it's getting activated. And this is not the CAR T cell, it's just a natural T cell that's getting activated to attack this cell. A couple of receptors are activated. And then the cell could inactivate the response if it has that yellow protein on its surface to say, no, don't attack me, I'm normally healthy. And so cancer cells can start expressing those immunosuppressive molecules. And then the last thing is that here's the cellular therapy type approach strategy, combined with also pharmaceutical therapy, where they have an antibody that can block that receptor and block it from turning off the response. So, modern medicine rolls on and on and on. They have some success and they're trying to fine tune it and advance it. And so it includes not just the cellular therapy, but some traditional pharmaceutical style therapy like that. And again, just to say this, right, that, this keeps rolling forward and scientists learn more and more and tweak it and adjust it to have it be more effective. Yes?
So did they add more genes into the CAR gene to make more receptors?
Yeah, so that's what they're doing, is that they're making at first, the first plan, like the first and second generation CARs were, let's take the 3 receptors and make 1 new CAR receptor. That'll be really sort of hyper excitable, right, like that. Then, that's too excitable. So now we're going to try to build a second CAR that will temper that and make it more sensitive so that the cells only attack in a really targeted way the cancer cell. So again, that's beyond our course, but Riley's going to be here on our last day of a course, and part of it is to tell the big picture part so you can pull all his work together, but part of it is a little bit like leaning out toward, what's the future look like. Okay, great. So I wanted to show this too, because for your studying, you want to go all the way back, right? And on the second exam, we were drawing exams and drawing genes like that, like we did all through our course. And you don't have to be an artist, but you need to be able to draw and label the parts of a gene like that. So I asked about that in some of those translocations, and some students were just not doing a good job drawing a gene like that. So it's going to go all the way back, and you're expected to know some of this stuff really well, like that. Like, I can draw a gene all the time. It's made of DNA. It's physical. And it's composed of 2 parts: a transcription unit that could have a coding region if it's a coding gene, or not, and then a regulatory region. And the cis regulatory elements are most prominently represented by a promoter that's right next to the start of transcription, because that's what the promoter binding transcription factors do, is they show RNA polymerase where to start, and then some upstream or other located but cis tethered element called an enhancer that powerful transcription factors will bind to. Yep, yeah, and I don't want to be too critical of that, because those same students that didn't draw a gene well in one place, they did a good job of drawing a cyclin gene and saying, oh, here are 4 different transcription factors that regulate that. So we just want to just be solid, you know, throughout the, you know, any questions about, yeah, okay, this is this, and it's physical, and you can represent it. And again, you don't need to be a great artist, but, you know, make some space, take some space, and do a good job. Okay, then I just wanted to talk to you about this, kind of looking beyond the end of our course, right? So here's Wikipedia, and Wikipedia is, I think, targeting sort of a college level reading, right, like that. Somebody that you know, is an expert in the field. And this is just the MIC Wikipedia page, like that. And we know something about MIC, right? So it just says, in cancer, MIC is often constitutively persistently expressed. This leads to increased expression of many genes, some of which are involved in cell proliferation, contributing to the formation of cancer. And we know why. Is because MIC is a transcription factor, and it activates a set of genes, like that, a set of target genes or response genes. A common human translocation involving c-MIC is critical to the development of most cases of Burkitt's lymphoma. Constitutive upregulation of MIC genes have also been observed in carcinoma of the cervix, colon, breast, lung, stomach. MIC is thus viewed as a promising target for anti-cancer drugs, okay? And so he talks about that. There are some genes that just are seem more likely to be more kind of vulnerable to mutations or misregulation and be oncogenes. Then there are some of those are more accessible to develop some kind of targeted therapy. So what about MIC? And it's unfortunate, this is Wikipedia going, like, unfortunately, MIC possesses several features that render it undruggable. That's one reason I showed this. It's undruggable. That seems pretty bleak, right? Like that, scientists are supposed to have that attitude, such that any anti-cancer drugs for MIC dysregulation will require acting on the protein indirectly, like that, such as by targeting the mRNA for the protein, rather than a small molecule that targets the protein itself, like that. So, you know, it's kind of sophisticated, but you could wade through that. Here's some more MIC discovery. The MIC family was first established after discovering homology between an oncogene carried by the avian virus myelocystomatosis vMIC and a human gene overexpressed in various cancers, right? So that goes back to, they found it first in the 1970s in an oncogenic retrovirus. What's the difference between a normal retrovirus and an oncogenic retrovirus? The oncogenic retrovirus has picked up a host gene. It's not the virus itself, it's the host gene that's problematic. And that was a foot in the door. And then they quickly left the viruses behind, because they're like, my gosh, that same gene we found in the virus in the 1970s, in the 1980s, we found it just in naturally occurring human cancers, like that. So really important concept, you know, that we've said over and over in our course. More MIC. The most frequently discussed example of c-MIC as a proto-oncogene is its implication in Burkitt's lymphoma. Blah, 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 blah. Cancer shows these translocations. It describes the translocation like we know. Then function. MIC proteins are transcription factors that activate expression of many pro-proliferative genes through binding enhancer box sequences and recruiting histone acetyltransferases. So it's telling us two things there. It's a transcription factor, and it's telling us how MIC stimulates transcription. And what does it do? It upregulates one of the possible rate-limiting steps, and that is chromatin structure. So it activates histone acetylases that are going to actively catalyze the addition of acetyl groups to histones and help the gene change from a closed heterochromatin that cannot be transcribed to an open euchromatin, like that. And that's true for all transcription factors, that each transcription factor has some specific way of regulating a rate-limiting step in transcription. And so we didn't talk about that in our course, but here it is, just Wikipedia just dropping it out like that. MIC is thought to function by upregulating transcription elongation of actively transcribed genes through recruitment of transcription elongation factors. whoops, and it actually does a second thing. Not only does it help open up the chromatin, but it helps release pause polymerase, like that. So it's two of the things that we talked about. So MYC is a powerful transcription factor that can upregulate genes that are needed for gene expression, like cyclin, right? I mean, needed for the cell cycle, like cyclin. And it's saying, well, MYC binds and it unfolds the chromatin of the cyclin gene, and it actually helps remove or push arrested polymerases on down the transcription unit. Yep. So pretty interesting just to see that. MYC is activated upon various mitogenic signals, such as serum stimulation or wind, sonic hedgehog, or EGF. EGF is a lot like PDGF, and we said that in that wound healing thing, we got PDGF activating RAS, RAF, MEK, ERK, ELK. It says by modifying the expression of its target genes, it says in parentheses here too, that via the MAPK, ERK pathway. That's how some scientists make a summary statement of the RAS, RAF, MEK, ERK, ELK pathway, as they call it, MAPK. By modifying the expression of its target genes, MYC activation results in numerous biological effects. The first to be discovered was its capacity to derive cell proliferation. That's what we really like. But it also plays, it says it upregulates cyclin, but it also regulates down up P21. So we didn't learn about that. That's okay. We'd be like, interesting, right? It's a repressor of the P21 gene and an activator of the cyclin gene. That makes it sort of a problematic gene, like an Achilles heel, where you feel like, ooh, you're really fiddling with things if you fiddle with MYC. And then it says, but it also plays a very important role in regulating cell growth, so not just the cell proliferation, but the actual growth of the cell. So it makes more ribosomal RNA. Let's make some more ribosomes in the cell like that. And it inhibits apoptosis, or actually it's saying it stimulates apoptosis in some cases, differentiation, and stem cell self-renewal, like that. Nucleotide metabolism genes are upregulated by MYC, like that. So it's talking about the various target genes, and that's something we want to make sure we have from our course, is that we show a transcription factor, and we usually talk about regulating a specific gene to get familiar with it. But most transcription factors regulate several genes. And MYC is a problematic thing because it regulates a bunch of genes that are dangerous genes, like that. Yep.
So you said that it
Yep.
deactivates BCL2, so
Yeah, so that's this funny thing, that one of the reasons we didn't talk about that regulation of apoptosis very much is because anytime a cell gets stimulated to go through the cell cycle, like PDGF in skin cells, that almost always involves also affecting the apoptotic pathway. And just the overall general idea would be that the cell needs to be sensitive to what's happening. When it starts going through the cell cycle, the cell should be like, am I doing the right thing here? What's kind of really confusing is that for some growth factors will stimulate BCL2 and some will repress BCL2, because there's a kind of balance that's happening. That this is not really saying you should go through apoptosis. It's sort of saying you should think about it, like that. And so that's one reason why we kind of went easy on apoptosis, is because of the complexity of it. That BCL2 gene family I mentioned creates a kind of like a majority rules, almost like a voting group of proteins. Some say yes, let's go through apoptosis, let's be pro-apoptotic. Others, like BCL2, say no, let's inhibit apoptosis. And it's a family, and there are many examples from cell to cell about which members of the family are expressed and what their reactions are, like that. So yeah, it's a little surprising to see that MYC decreases BCL2, but it's not really inhibiting apoptosis. It's just saying, I don't think you need to go there. Yep.
So the last sentence in this says necessary for MYC-induced proliferation or cell growth.
Yeah.
Isn't it PDGF that's signaling the cell to grow and not MYC?
So yeah, that's a good question. MYC is, and some of these growth transcription factors that we learned about, like ELK, they are controlled by upstream factors. And some of them are from outside the cell. So that's what we get going on, where it's really in our skin cells, when they're healing, it's really PDGF that's the factor that's stimulating it. And then RAS, RAF, MEK, ERK, ELK, MIC, and so forth are downstream of that. They're on that pathway to the response. And then beyond that pathway, well, they induce cyclins, and so cyclins are needed for the CDKs, and we start getting that pathway going like that. So it's a long pathway within the target cell, and MIC is sort of on the middle of it, like that. Yep. Yeah. Okay. So I wanted to show that, and then just connect that to this. That's all. MIC is all cancer talk. Cancer, cancer, cancer. And that's because there's a lot of research on cancer, because it's, you know, just about the leading cause of death in the United States, like that. Really a big challenge. And a big sprawling challenge, right? Heart disease, that's a big sprawling challenge too, but it doesn't just go on and on and on, because there are not many, many different kinds of heart disease, like there are many, many different kinds of cancer, like that. So MIC fits into that. That's the basis for our course. At this point in the course, we also want to emphasize that MIC also plays a normal, healthy role. And some of the proteins that we've talked about, they have a role in cancer, they have a normal, healthy role, and then they'll have a variety of roles in disease that are other than cancer. Like some of these proteins play roles in Alzheimer's and other completely different disease states, like that. And so, you know, it's important that our focus has been on cancer, but we're introducing principles of a lot of different diseases and healthy states in humans. So I just wanted to show you this. How many diseases are gene related? So it says about 10,000 human disorders are involve misregulation of genes. So, you know, it's not like we're just having a course on cancer. We're using that as a vehicle to introduce you to these molecular principles that are relevant for thousands of different human medical conditions, and then all of our normal, healthy state, like that. Then it's true that this also is introducing the concept of regulation of genes for all organisms, every organism on our planet, like that. And so again, you know, we kind of went with this targeted focus to learn and put it in a context like that, but it's just a very broad introduction. So I said this, I just wanted to point this out as we're, you know, in the last week of our course. So why is this a good course for a science major? Well, because it's cancer biology, and everybody is kind of tuned into that and should want to learn more about it. It's about our friends and family and our own bodies, and you know, the likelihood that we're going to get cancer, and our friends and family and our pets will. So that's a good reason. And then it's an entry into understanding molecules in health and disease in humans. So another medical reason. Then you just find orthology in all animals in many cases. And so all animals like FOXO. How does any animal grow old, including the yeast that causes bread to rise? Oh, FOXO plays a big role. The orthologs of FOXO are found even in the yeast that causes bread to rise. A single-celled eukaryote actually grows old. You know, it's just stunning, right? That is a big part of the story of our course, is we can talk about cancer. We're really talking about lots of different human disease states and lots of human health and all kinds of stories in eukaryotic biology. Then it's all animals, or all metazoan, all plants and animals, like that, or all eukaryotes, like that. It just ranges just tremendously, and so that was a big emphasis. So we're learning about molecular mechanisms that are shared, you know, really widely. And you know, it's a focus on eukaryotic cells and not bacteria, but a lot of these stories could be taken into bacteria as well. And so it's interesting because, you know, you're more familiar with things like tissues and physiology, like that. But, for example, how much more significant is MIC in your life than your heart? Oh, a lot more. Yeah, because you just wouldn't have even shown up without MIC, like that. So it's a weird thing because fruit flies don't have hearts or bones, like that, but they have so much similarity to our body that you can use a fruit fly to study human hearts and bones. Because down at this molecular level, there are orthologs that are regulating other tissue development in fruit flies, even though they our last common ancestor did not have many of the tissues that, didn't have, our last common ancestor between us and fruit flies did not have a heart or any kind of circulatory system at all, like that, So some of the biology that you've learned and, you're really enthusiastic about is just, way outclassed at this more foundational level by these topics. Then the last one, and the one I just wanted to emphasize, because this is what I really like, is it's physical cause and effect relationships that are just based on evidence. Just like boom, 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 like that. And so that's good learning, that's good learning just in the experience of learning how to learn. Because it's just like, how am I going to deal with all this stuff? How am I going to organize it? So it talks about that too. And, folks in 100 years ago, they used to take courses in like Greek and Latin to learn about learning. And this is the modern equivalent of that, of just good and healthy learning, okay? So again, it's our whole course we're dealing with, and then this is just a couple of articles I found. This is just an article about that woman who had skin cancer, lots of research on that specific, that's the name of her skin cancer, right? Very odd, rare skin cancer. Here's a article, scientists working on that B-cell non-Hodgkin's lymphoma, like that. Okay, so we're starting to pull this together, and hopefully having you take it with you out into the summer and beyond. So chapter 11, the nucleus, and it's organized into these 3 parts, and they're ranked in order of number of words in the thing and how much we have to say about it. So we have very little to say about nuclear bodies. There'll just be a little bit to talk about inside the nucleus, and there's some regional localization of activities inside the nucleus. For example, the nucleus is just a big, sphere, and transcription occurs in the nucleus, and it especially occurs in certain locations. And so people can refer to that as a nuclear body. Localization with the nucleus, and they call it a transcription factories. So we'll look into that a little bit. But there's just a little bit of that about the nucleus and what's happening inside of it. Well, transcription is, and what can we say at the cell biology level? And that is, it's organized. It's not just one gene at a time; they're clustered. Then, at a higher level, we'll talk a little bit more about how the how the chromosomes are organized. And I think all of this is in talking about G1 type stuff, G1 and S phase, when the cell is really doing its living. So organization of the chromosomes, that's when the chromosomes are invisible to our eye because they're spread out inside the nucleus. And that's when they're really doing their function. During G1, they're being actively transcribed, and we're seeing genes turn on and off. So how is that organized inside the nucleus? And then during S phase, the chromosomes are still invisible, but they're getting replicated. So how that's organized, like that. So in addition to transcription factories, people talk about replication factories. So regions within the nucleus that are really in charge of regulating S phase. And that's where the MCMs and all those guys would be piling in to get that organized. And there's some subcellular organization of that. And because there's some subcellular organization, there's some regulation. And we aren't going to dive into that because it's really an advanced topic, but just to show you that some of our molecular stories fit inside the cell and inside the nucleus a lot, right? The first one is the one we'll talk most about. So the nucleus is surrounded by a membrane, and traffic in and out of the nucleus goes through little holes, little nuclear pores. And that's a good example of cell biology. And we've talked about those nuclear pores some without naming them. For example, on the first day of our course, we said the FOXO transcription factor needs to get into the nucleus to do its job. And the AKT can phosphorylate FOXO, and 1433 will bind to it and prevent it from getting into the nucleus. So we'll find out what's the detail of that. Well, it's not able to get through the nuclear pore. Why not? Right? All the transcription factors we talked about, their genes are inside the nucleus, so the gene has to be transcribed. The RNA needs to get processed. It needs to leave the nucleus through a nuclear pore. Then it needs to be translated into protein in the cytoplasm. And then those transcription factors have to get back into the nucleus for their job to get done. So there's a story of export and import through these nuclear pores, and that's the thing we'll say the most about in our chapter. It matches that too. And it's pretty straightforward. There's some structural stuff, and I'm just going to cut and I'll back up to this, but I'm just going to cut to this just to show you this, because it's a summary image. Okay? So again, we'll introduce this, we'll back up, but this is what we're heading for that first part about the nucleus and the nuclear pores and the nuclear membrane, is that within the nuclear membrane, so this is the bag that the nucleus is, there are these holes called nuclear pores, and they're pretty big, and they have a pretty specific structure, like that. And stuff needs to get imported, and stuff needs to get exported, like that. Who needs to get imported? Well, proteins like transcription factors, RNA polymerase, DNA polymerase, those nuclear lamin proteins that are part, histones, the proteins like histone acetylase that modify it. Lots of those guys need to get into the nucleus. They need to go in through the nuclear pore, and all those proteins have a tag that is a tag they are required to have to get into the nucleus. So it's a regulated step, like that, importing, like that. And then lots of stuff needs to get out of the nucleus. I mentioned the mRNA. When a gene gets transcribed, the RNA is inside the nucleus, and it gets processed and it gets sent out of the nucleus, and so that's exporting. Okay? And so from this summary, what we get from this summary is that those molecules need to have tags to identify them and where they need to get to, and there's some proteins that recognize the tag. So there are helper proteins that help all these transcription vectors get into the nucleus. And we're going to learn the name of one of those helper proteins, and because it has a good name, it's called important. There's a bunch of them. Our book has a table that lists a few of them, and important is the one we'll learn. And you see, here's what important does. It's a protein, and here's a protein that is represented as needing to get into the nucleus. Important binds to it and helps it get through the nucleus. And it just is doing that by like being a flag and saying, we need to get into the nucleus, here we go. So important goes into the nucleus with its cargo, the transcription factor, the histone, whatever it is, the protein it needs to get in, and then it lets go of it. And then important gets exported. So it's on a little cycle of bringing stuff in and then getting back out so it can bring more stuff in. Yep.
What is that one with the GTP that's with the important?
Like in here?
Yeah.
Yeah. So it's a cycle. So it's a cycle like that. We're bringing stuff in, and then we're getting the important back out. So we're recycling it. Just like if you buy milk in a glass bottle, you buy it and you have to bring it back. You're going to bring it back and refill it and use it again. So that's what this important is doing. And the thing that Brooklyn just asked about is that in this step where we just need to get it back out, it actually requires energy. It requires hydrolyzing a GTP to get it back out. And the reason the book is saying that is because this cycle is it's like an information regulated process. So it's like a gatekeeper that's not just letting stuff flow in. So transcription factors like FOXO are not allowed to just flow in. It has to have a tag, and this important needs to bind to it and bring it in, and then we recycle important. And on the recycling pathway, there's an energy requirement. And what that does is that energy provides the switch-like flow. So it's a little bit like a light switch, where to get a job done, you actually have to push on the light switch. And it's a little bit of a difficult concept, but what it's showing is that for this to be a regulated, information-dependent step, you need to have the process somehow be requiring energy. It's kind of like a little teeter-totter notion, like that, where, well, if you want the kid to go up on the other end of the teeter-totter, you're gonna have to use some energy. So it's not sloppy, it's regulated. The kid will not go up without it. And that's why the book, one of the main reasons the book is trying to show that. And it's curious to see that, you know, importance really doing its job here. But to get that job done, you need to get it out. And that's an energy dependent one. And so that makes the whole process energy dependent and like that thing where it's not sloppy. It's a switch-like event, and the switch event is here. But because there's energy in the pathway, the overall pathway is energy dependent. So we have a couple questions about that.
So, to stop importing from bringing stuff in, you would like the energy be regulated to.
Yeah, that's a good question. You know, you could regulate, you could adjust this importance function at steps along this pathway. It just requires one step for it to be energy dependent. Yep.
So I assume like this is happening in like G1 and S phase. So does important tend to hang out in one particular area over the other? Whether
I don't
it's
know. Yeah, like where you see most of it. Is it mostly hanging out in here or waiting to get out, or is it mostly up there? I don't know. There would definitely be pools, and I don't know if one would be kind of prominent. Yep.
Gotcha.
Is important considered a chaperone protein?
No. So Eva asked about a chaperone protein, and chaperone proteins are proteins that help other proteins fold. That's the first thing you'd say about a chaperone protein. It's not simply bound to it, it's actually helping it fold. So heat shock proteins are often chaperones. Yep. Yeah. So there's that. Now, just as this analogy, right? So when I was a kid, I had these little toy cars called Hot Wheels, and you put them on a track and let them roll down the track, down the hill. And when I was a kid, they came out a new product, which was that there was a thing that was just spinning, and so now you didn't have the Hot Wheels roll down the track. You could just make a circular track, and the Hot Wheels would roll through this spinner, and it would like push them, and they'd go all the way around the track and come back to here. And so that's a little bit like what this analogy is like, is there's a track, and for important to go around the track, you have to give it some energy. And if you don't, you can't get that work done. That's actual information work to get that in. So it's a hard concept, but if you start to grasp it, it's really satisfying to go to this one, because this one's more direct, and it's satisfying. So think about this one, and then, remember all those kinases that we've studied? Every kinase is an enzyme that has catalyzing a chemical reaction, and the chemical reaction has substrates, and the substrate will be a target, like that, you know, where it's going to phosphorylate a target, like AKT is a kinase that will phosphorylate FOXO. But it also includes another substrate in that chemical reaction always, and it's ATP. So the phosphate the kinase gets to put on any of the targets we talked about is from ATP. And as a result, there's an energy flow in all kinases. And that energy flow creates a switch so that the activation of that target protein or the regulation of that target protein, whether it's P53 or P27, any of those, is a switch-like thing. Again, back to the light switch. Light switches are crisp, like a teeter-totter. It's in one state or the other, and the way to get that crisp one or the other state is to have the light switch dependent on energy. You need to push it, to flip it, like that. And that's what a kinase is doing, is its target has two conformations. And they're not sloppy, it's in one or the other. It's either inactive or active. And by phosphorylating it, you switch the target to the other conformation state. How can you do that? How can you design that so it's crisp, like a switch, and not floppy, where the target is just on or off sometimes? And the answer is make it energetic. It's weird, because it's a kind of a contrary use of metabolic energy. But information flow in biological systems is energy dependent, and that's what makes it information flow. So 2 examples for our course. One would be this important process, which is a curious like cycle, like a car racing around a track that gets pushed once around the track and goes the whole way around the track. Or just like the kinase that is switching the target from one conformation to the other, like that, okay? So that's that, important. And all of the import is involving proteins like important. It's all energy dependent. It's a gatekeeper. It's like an information flow. You can't get in here like that, crisp, like that. Then the other thing is just to mention this, and that is that for, I guess it's going to be, we'll come back to this. But then for,
So it kind of acts like the cover charge to get into a bar.
yeah, yeah. I really like the light switch, because the light switch has really 2 conformations, on or off. And you don't want that to be sloppy. We don't want it to be in here and the light's just flickering on and off. We want it on or off. And both states are important, right? Off at night, we don't want to be wasting electricity with it just flickering. On, fully on, like that. And all these targets of kinases have 2 conformations. They're a little bit like a teeter-totter, and it would be kind of a sloppy thing where they'd flip on and off unless you had a crisp way of really one or the other state. Okay, so here's the other protein that we're going to learn about, and that it's called exportin. So that's great, right? So here's exportin, and it's just showing that it's taking stuff out. And what it's showing is that it's going to help take some cargo out, and that can include RNAs like that. So the book talks about that a little bit. There's a lot of detail in these because it's cellular biology. So they'll show, physical location, like the nucleus, the nuclear pore, and so forth. But we're really interested in that molecular pathway. The exportin also is energy dependent. So there's a step in that recycling pathway. So in the case of exportin, it actually binds to GTP, and it doesn't hydrolyze GTP until it gets the cargo out into the cytoplasm. So the details of the timing of the hydrolysis of the GTP is not a big deal. It's just that it's involved in that pathway. To keep that pathway going, we need to keep inputting energy. Yep?
Would you say that there's like a confirmation change of the helper protein as well? Were you saying that or not?
Probably, because the helper protein is also probably changing states, because it's probably going to change to bind the protein and then change to release the protein, like that.
Okay.
Yeah.
But the big takeaway is more just like it needs energy to keep going in a cycle.
The big takeaway is that information flow, that's what this is. It's gated, it's information. You're not getting in the nucleus. And how do you control that? How do you mean to say that we're going to have this information flow? And the answer is, it's going to require energy to make it, one direction, and to make it consistently go, out. You need to have an input of energy on that pathway, like that. Yeah. Yeah. So it's interesting to see too, this concept, recycling, I know that, like getting the milk in the bottle. Yeah. There's a lot of that in biology and biochemistry, because you can put stuff on that recycling pathway that allows that whole pathway to work. And it's a very simple pathway, so that's one reason, to focus on it. So we have that important and exportin story, and that's really the summary figure that we want to get to. And we want to back up and talk a little bit about the nuclear pore and just, its location and stuff like that, and then a little bit about what's going in and what's coming out. Like how does important know what to take in, like that, okay? But we're going to that, and like I said, and the book shows several of these figures, because there's a lot of stories going on here. We know that one about FOXO. FOXO can't get into the nucleus because it gets phosphorylated and bound by 14:3:3, like that. The book is introducing other ones, partly because it's talked about those in the textbook that we didn't read and stuff like that. So you don't need to learn new additional examples. We'll go to some of the ones we already knew, and we'll plug our stories into there, right? So we know about heat shock transcription factor and the estrogen receptor and FOXO. We have some examples of transcription factors that are getting into the nucleus like that. So we'll look at them, okay? So here's just that table that says here's some examples of these proteins that are playing a role in getting things inside and outside. They're the proteins that are carrying that cargo in and out. And the only one we need to learn is important and export, like that, from that list. So I'm not going to be like, oh, hey, what's another one? Like that. We're targeting that, learning like that, okay? So let's just back up and say, the book shows this again, not surprising, it shows some electron microscopy because that's the birth of cell biology, where they looked at the nucleus and they're like, let's zoom in on that and see what's going on. And they see, under an electron microscope, they see the nuclear pore. The nuclear membrane itself is a double phospholipid bilayer, so the cellular membrane is just one phospholipid bilayer, the nuclear membrane is a double one. And it's kind of interesting that the nuclear membrane's outer membrane is just part of the endoplasmic reticulum. So there's a lot of really interesting cell biology that we could get into with regard to the structure of the nuclear membrane and the nuclear pore in it. And so, images like this, and they're just emphasizing the main principle of how the nuclear membrane is really tied to some of the other organelles, like the endoplasmic reticulum, okay? So then another electron micrograph like that, so they're like, back in the 50s, they're like, oh, crap. This guy, Keith Porter, was a scientist that was just looking and being like, oh, look at that. That looks like a pore of some kind. Let's call it a nuclear pore. They'd never seen it before. And they had no idea what's that about, like that, right? So just a kind of an explosion of questions from these electron micrographs. And that continues today. And of course, cell biology is not limited to electron microscopy. So here's something they want to talk about. They want to talk about the nuclear lamins. So that's something we learned about that comes up in this chapter. And those are those proteins that are on the inner surface. And so here's the, just zoomed in on nuclear pore. And what would we want to know about it? Well, there are nuclear pores, and they're the way you get in and out of the nucleus. Nucleus has a double phospholipid bilayer. Nobody's going through across that thing. It's quite a significant barrier. Everything's going to go in and out through the nuclear pore, basically, like that. And then, see these yellow things? Those are nuclear lamins, like that, say right there, nuclear lamins. So the shape of the nucleus is provided by those nuclear lamins. We learned about them because they get phosphorylated by CDK1 during M phase, and in many cells the nucleus breaks down, like human cells. Then it's just also showing that, although invisible during interphase, the chromosomes are in there. They're inside there. And some of them are attached to the nuclear lamin, and there's interesting stuff going on there. And then it lists some proteins that play roles in helping organize the what they call the cytostructure, the cellular structure, but we don't need to learn those, like that.
So the yellow like crisscrosses the
Yeah, those are the nuclear lamins. So we know about that. We know about chromatin and these chromosomes. And just the idea that there's some structural shape. We'll learn a couple of concepts about the organization of the chromosomes, but we don't need to look at this in detail. So that happens when you see in some cell biology, there'll be a figure like this, and it'll show physically the layout. And there's plenty of space to introduce a bunch of different topics in this one figure. And so we want to wade through that and get to what's significant for us. And what's significant for us is that things are going in and out of that pore, like that. And they need help to get in, and the help that we're learning is important. They need help getting out, and the help we are learning about is exporting. Okay? So again, they're showing like there's this cytoarchitecture here, like there's this structure like this. The nuclear pores are really a pretty big biological complex with a lot of structure to it. That's what's shown here. This artist is drawing it like what you can see on your electron microscope. The hole itself also has some material in it like that. So scientists have a lot of work to do. They've been working on it for a long time. And another kind of image showing those are the nuclear lamina, the yellow, and then the nuclear pores. Great, like that. Then it says this, and just a little sidelight to say this. that seems really dry, like, okay, got the structure going on and stuff like that. And it just does stuff like this, because it's just fascinating to see this, and back to these genetic diseases. So here's a child who has this disease called progeria. It's an inherited disease, a genetic disease, and the child is growing old really fast. The child is like, has a chronological age of maybe 7 or 8 years, but they have literally the body of a 90-year-old with all the kind of medical conditions of somebody who is that old. And you almost for sure have seen that, on TV, the news, that around the world there are a few children that just age really, really rapidly. So in this progeria, this child has some kind of mutation that's causing their entire body to age really fast, And, we could, place bets, bet a beer or something like that on what you think it would be, right, from our course. And, probably Foxo is messed up, huh? Something like that, or some regulator like that. And the answer is these children have mutation in the nuclear lamina protein. And scientists are just like, holy smoke, no clue, right? It's just considered a structural protein like that, just like it helps give the nucleus its shape. And that's just a really good thing, so a little bite for us to look at, because we breeze through this image going, yep, great, great, great. But there's just a lot of biology there. Some of this, and again, we don't need to learn about it, but some of these connections and stuff like that are somehow really important in the rate of aging in humans. And I don't think anybody has a suggestion. Like, what would that be? What's going on there with the children that have that? They're aging too fast because they have some problem in that what looks like a structural protein. So that's just in there, and I think that's nice, because it's true. But it's also just being like, oh man, there's just so much more biology for us to understand. Okay, all right. So then, just back to this, and now we're going to talk a little bit, and we won't have concept chats this week because we'll just be, working on wrapping up like that. But I super encourage you to learn by recalling. That's what we're trying to do with concept chats. And recall by, chatting with other students or friends and family, and explain that to them, right? That's how you learn stuff, is by recalling it. So go home and say, you saw that progeria thing, or Google it and say, what's going on with that kid? It's really strange and surprising, okay? So there are some small molecules like water and some small molecules that can get in and out of that nuclear pore, just through passive diffusion. But most of this stuff is going to be transported through the use of proteins like important and exportant. And so who we spend a lot of time thinking about who needs to get in are proteins. All the proteins that function in the nucleus are synthesized out in the cytoplasm by ribosomes in the cytoplasm. Now they need to get in to do their job. So all those proteins we talked about. Then the RNAs that get synthesized, transcribed in the nucleus, they have to get out of the nucleus to do their job, like that. So here's the story about how proteins get into the nucleus. And the answer is all the proteins that get into the nucleus, with extremely rare exceptions, have a little part of the protein that is got several amino acids with a positive charge, and it's referred to as a nuclear localization signal, or an NLS. So what proteins have that? Well, FOXO and MYC and every transcription factor that we talked about. Histones have that NLS, DNA polymerase, so forth. So lots of proteins have the NLS, and that's just the tag. So important binds to proteins that have an NLS. What's going on with FOXO? When FOXO gets phosphorylated, 1433 binds to it and covers up the NLS. So we can just draw a little pictures of that. Here's another, this got turned off. Here's another example of that we could look at that's cool to see. And it's just a little add-on to, we talked about the heat shock transcription factor in our course. And we learned that heat shock transcription factor is present in every one of our cells all the time. So always expressed. in every eukaryote. Like that. So, yeast that causes bread to rise, right, like that. And it's present as a protein monomer. And then when there is heat stress, when heat stress occurs, that protein changes shape. So where there's a stress, it could be heat or some other protein stress that causes some denaturation, protein denaturation. That the protein changes shape, so now it changes shape somewhat, and it exposes a trimerization domain, and so now, after stress, the protein forms a trimer. And that binds to the enhancer of heat shock transcription of heat shock genes, because the individual monomers have very weak affinity, but collectively the trimer has like 3 hands that it's using to grab the DNA, and so now it's a good transcription factor. An additional detail that we didn't talk about before is that the heat shock transcription factor also has a nuclear localization signal, or an NLS, and when it's a monomer, the heat shock transcription factor is in the cytoplasm. Its job is the nucleus, but it's not doing its job because it's a monomer, so it wouldn't bind very strongly. But also, it hasn't even gotten to the nucleus, because when it's a monomer, the NLS is covered up. And so important protein doesn't bind to the monomer. So the activation of heat shock transcription factor is just to change the folding, and that has 2 effects. 1, it exposes the trimerization domain, so we get trimers. 2, it exposes the NLS, it's so important, takes it in the nucleus, like that. So it's kind of a double level of regulation. The transcription factor is there, just waiting to be activated. As soon as it's activated, trimerizes and gets taken into the nucleus by important. So there's important not binding, and then when stress occurs, the important binds, because now it detects those nuclear localization signals, and it transports it into the nucleus where it can do its function. So like I said, the book rambles through some examples in chapter 11 of like, hey, here's a protein you need to get into the nucleus, and it's picking ones that it's kind of talked about in some chapters we haven't read. We didn't read, but here's one for us that's a really satisfying one, like that. Okay? Okay, like that. Okay, yes?
Is it important always binding to NLS, like that? Just like how
Yep, that's how proteins are recognized to get into the nucleus. I don't know of any exceptions. There are always some exceptions, but it's the thing. Is if a protein's going to ever get to the nucleus, it's going to have an NLS. And important, or one of its relatives, will bind and haul it into the nucleus. Yep.
and it's able to be exposed after the denaturation?
Yeah, the stress, it changes the shape of the heat shock transcription factor, and it exposes both the trimerization domain and the NLS. So it trimerizes and gets hauled into the nucleus where it gets to do its job.
I know space is of importance within the nuclear pores, so is the HSF protein pulled into the nucleus via important prior to trimerization or after?
I don't know, and I don't know if anybody knows that. I think they're so close that, yeah. But one thing is that if you let's just talk about this. So we have some time here, let's just talk about this a little bit. So you could test this idea a little bit, learn a little bit more about it by doing this. You could take some cells, either unstressed cells or stressed cells, so you'd have 2 populations of cells, and what you could do is you could isolate the nuclei, and then you could ask, where is the heat shock transcription factor? Is it present? And if so, where? So let's just remind you of this because our final exam covers the whole course. So way back in chapter 1, we learned that scientists could use a centrifuge to separate cells from liquid. So you can use a centrifuge and the cells would pellet down here. Then we learned that you could break up the cell, so you could lyse the cell or break it open, and then you could use a centrifuge and you could separate out parts of the cell. So we could do this, and instead of pelleting cells, we could lyse the cells and then centrifuge, and we could isolate nuclei away from the rest because the nuclei are big, so they would still pellet in a centrifuge. So the steps are to lyse the cells and then centrifuge them and then save the nuclei that are in the pellet. And what you could do is you could say, in either this liquid or the pellet, where's the HSF, the H-Shock transcription factor? And in unstressed cells, the H-Shock transcription factor is in the cytoplasm because the nuclear localization signal is covered up. And so we would find HSF in unstressed cells in the cytoplasm. And in stressed cells, the nuclear localization signal is exposed, and so we'd find it in the nuclei because it's been translocated into the nucleus by important. So that's just an example of reaching back into chapter 1, because the book was introducing some molecular techniques there, chapter 1, but also some cell biology at the end of chapter 1. I was talking about centrifuges, and cell biologists had used centrifuges a lot to isolate some of these components, right? They first saw the nucleus or the mitochondria under an electron microscope, and then they're like, we want to isolate it. We want to break the cell open and isolate some of these organelles so we can study them individually. The last thing is, you might remember that we briefly talked about this. When we were lysing the cells, we could use a detergent to gently lyse them, but we want to be careful not to lyse the nuclei because they have a membrane too. And the instrument that's used to lyse them and prepare nuclei is called a Dounce homogenizer. And a Dounce homogenizer looks like this. It looks like a test tube that has a bulb, and then it's just a test tube. It's made of glass. It looks like this. And then inside of it, there's like a stem with a handle and a glass ball. And the user holds it here, and they move this glass ball up and down in that. There's out here, because it can hold more liquid if it's flecked out. Right here, if we zoom in on that, the ball of glass is made so it's got a very specific gap with the cell, with the test tube. There's just a little gap here, and a little gap here, and that gap is narrower than a cell, but wider than a nucleus. So that's a nice thing to tie this in to the very first part of our course. You can use a Dounce homogenizer to isolate nuclei. Just take the cells and put them in a little bit of detergent to help it break it open, and then just go like this a couple times, and it just physically tears apart the cells that get caught between the wall of the Dounce homogenizer and the glass ball. They get torn apart, but the nuclei slip by. So you take that, and then you take that nuclear extract, and you centrifuge that, and the nuclei will pellet, and all the material that was in the cytoplasm will stay in the liquid, like that. And that's just an indication, a little bit of like cell biology. So looking under a microscope, see stuff, they just try to isolate different parts of the cell like that to study that. Oh, we have purified nuclei like that. Yep. Okay, so we, I think, have to stop here. Yeah. Well, in a minute, but we'll be stopping here. And, I'll be handing back the exam. So with the link today, I'll send you the answer key video. And so with this, you don't get very much information. I just have marked stuff wrong and given you a circle score. then with the link today of the recording, you'll also see the answer key video link, and that will show you all the information